So hello everyone, I'm Eva, and on behalf of GenScript, I will be interviewing today iGEM Goethe University team. So the team, please introduce shortly yourself and tell me something more about your project.
Okay, so maybe I'll start. I'm Olivia. I'm the one that you were in contact with. I'm one of the co-leads of this year's iGEM team of Goethe University. Next to iGEM, I do other stuff like studying. I study physical biology, cell biology, sorry, in my master's program. And I brought you some of our heads. First of all, our wet lab head, Caro, and our IT head, Christian. And maybe Caro, you can start.
Yeah, hey, I'm Caro. I study biochemistry in a master's program in Frankfurt, and I'm leading the wet lab for this year's iGEM team.
So hey, I'm Christian, and I'm studying bioinformatics. And yeah, as Olivia already mentioned, I'm leading the dry lab team this year.
Great, nice to meet you guys.
Nice to meet you too.
So please tell me something more about your project.
I can start. It's maybe from what you've heard already from our emails, we're going to tackle antibiotic resistance, and what we're trying to do is to have something very simple to use and to detect antibiotics. And maybe I will ask Caro to continue because she's by far more skilled than I am in this. So you can go on.
So the basic idea is to use a whole cell biosensor. The advantages of whole cell biosensors is that as soon as you have it, it is quite cheap and easy to use. So cells reproduce themselves, and you can just use them to detect something in the environment. And these whole cell biosensors are always a transcription and translation based system. So when you have like a signal from outside, like an antibiotic in wastewater, and you have it in contact to your biosensor, you will have a membrane protein in our cases, which will then transduct the signal over a transcription factor so that in endogenous cases, an resistance gene is produced. And you can utilize that by replacing the resistance gene by a reporter gene. And the reporters we chose are on one side the beta-galactosidase. This is quite handy because the beta-galactosidase can turn white substrate into a blue substrate. So you have a direct visible change. You don't need a whole lab equipment to read out the results of your test. And you can get quite a fast and also easy readout of your results. And the other reporter we chose is GFP or any kind of fluorescent protein. And for those two systems, we want to make them applicable by putting them for the beta-galactosidase on a paper strip. So this can be like a portable COVID test, which you can bring anywhere where genetically modified organisms are allowed. And for the fluorescent proteins, you can put them in microencapsules like alginate bubbles. You might know them from bubble tea. So you can put them in a safe environment and test the solution you want to test.
Yeah, and maybe before we get into what else we're going to do with all of this, the idea for this project came from the growing danger from multi-resistant antibiotics. We have some groups that are working, multi-resistant bacteria, I'm sorry, some groups that are working on this. For example, Acinetobacter baumannii, I don't know if you know this strain. It's a really, really dangerous strain, and we will have more of those in only a few years. Like in 2050, we did some research from the WHO that in 2050, the deaths due to antibiotic resistance will rise to a very dangerous level. So we don't only want to detect one antibiotic, we want to do some more. And maybe Christian, because also he's a lot more skilled than I am, maybe you might want to introduce what we're going to do afterwards.
Yeah, that's a good point. So as Olivia already mentioned, we would like to have an portfolio of several tools that we can detect antibiotics, and several antibiotics, not only one. And especially we are focusing on beta-lactams, and that's where the dry lab comes in. And what we are trying to do is using mutations and so simulations by using mutations, and in that manner, we would like to mutate our receptors so that they are recognizing other antibiotics, and that we are going to confirm or verify using common simulations. And in those simulations, we are just interfering those physical principles which are applied to molecules. And that's where math can start, and we are then simulating or performing such an molecular simulation in that way. And in the end, we have then multiple, and in the end, we have then multiple receptors which we can use for specific beta-lactams or also other antibiotics.
We have the same idea, because the idea is to have a whole spectrum of paper strips. So it's very easy. Everyone can use it. You don't need special training, because right now all the methods, you need special chemistry training, special biology training, all these trainings that not everyone has. I mean, not everyone studies this kind of science. So yeah, this is the project.
Okay, sounds really interesting. And I have to say that the topic of rising resistance, it's getting more popular. But I'm also aware that it's not that common knowledge to most of the population. We know that overusing antibiotics is pretty popular, but we also don't, I think society is not aware of the contamination of antibiotics in environment. So do you have any applications, do you do anything to raise this awareness in society?
Yes, very much. I mean, in the future, we plan a lot more, but for now, we were at the fair at the beginning of every semester. There's a fair for all the first semester students. And this was the first time we tried to educate people. We had like self-made posters and all these. It was in the beginning, but still there were a lot of people very interested in the project, very interested in iGEM, but especially very interested in the issue that we're trying to tackle. And just lately, it was a few weeks ago, there is also a fair/festival at the university. It's called Night of Science. So I invite you next year, because it's really nice. And this is where we had our first professional talk. Me together with Carol, we talked about what would happen if we overused antibiotics even more, or what would happen if we kept on with this method to tackle bacteria instead of using other methods. And it was interesting how many people, even far away from this department, asked questions, and how they could be safe, and how they could change their lifestyles and all these things. And in the future, we're also trying to have meetings with students from schools, and have talks there, maybe do little workshops, and teach the younger generation, so they will have a brighter future when it comes to that.
Yes, definitely. That's good to hear.
Yeah,
I have to say that you also answered some of my questions that I had to you. But yeah, one more thing, because I'm very curious. You mentioned that you want to make this device very accessible. And you also mentioned that you want to make it sensitive to different kind of antibiotics, right? But how sensitive it would be, like, and what quantity of antibiotics has to be environment that you will see the positive result?
I don't know, maybe I mean, I can start. We're testing prototypes right now, and of course the wish is to detect traces. And we're trying to make it as sensitive as possible, and this is actually the aim, to make it as sensitive as possible. But I mean, we're still prototyping, so we're hoping for the best. And we've had some results. They're promising, but I don't want to say more, because I don't want to jinx it. But right now is really a good time.
Sure. Yeah, you should leave the most important information for the rest of the contest.
Yeah, because let's not get too excited, but because once it works, let's get excited then.
sure. Yeah, okay. Do you have any more information that you would like to say to our audience?
Yeah, maybe if we want to use another system apart from wholesale biosensors, we would have another chance to like don't use the transcription translation-based system, but to use the readout of the sensor proteins we are using directly. So what we can do, one of our proteins is a membrane protein, which is a metalloprotease. And proteases, of course, cleave peptides and proteins. And this protease has a very specific cleavage site. And we could also use a synthetic peptide, for example, which is modified by a quencher and fluorophore, and place these around the cleavage site. And as soon as we have a beta-lactam antibiotic which activates our protease, we can also detect this cleavage signal as there's no longer a proximity between the fluorophore and the quencher. This, of course, would require lab equipment and wouldn't be as accessible as the paper strips we want to also test, but would be a faster readout and maybe also an even more sensitive one. So we don't only want to base our detection on this wholesale approach, but also use like more advanced approaches to tackle this issue.
Yeah, got it. Thank you so much. So yeah, thank you so much for interviewing with me and for your time. And on behalf of GenScript, we really wish you all the best luck during the competition, but also during GenScript sponsorship competition. So yeah, good luck and thank you.